In this session, we will talk about the lymphopoiesis in detail. First, we will have an overview, and then we will discuss the process of lymphopoiesis of B cell or B lymphocytes in detail. Lymphopoiesis is the production of new lymphocytes from the bone marrow. This is the bone. Inside the bone, there is a bone marrow, and inside the bone marrow, there are stem cells. These are called pluripotent stem cells. In the production of lymphocytes, some of the pluripotential stem cells become early lymphocyte progenitor and then lymphopoietic stem cells, which are also called common lymphocyte precursor. To become B cell, it differentiate into Immature-B cell in bone marrow and become antibody-secreting B cell or plasma cell in lymph nodes and spleen. But let's move into detail that how lymphopoiesis of B cells are different from T cell, as both have common lymphoid precursor in common. The differentiation of B lymphocytes in the bone marrow can be followed by looking at the expression of different molecules by the developing B cell. One of the first signs that a cell is committed to the B cell lineage is the expression of a surface protein called as CD19. The cell, at this stage, is called pro-B cell. The cell then expresses the IgM heavy chain in its cytoplasm and is known as pre-B cell. The next stage is called an Immature-B cell, which expresses immunoglobulin M on its cell surface. Finally, the cell becomes a mature B cell, expressing both immunoglobulin M and immunoglobulin D on its surface. This B cell leaves the bone marrow and enters the bloodstream. It is during this process that unique molecular mechanisms operate on the immunoglobulin genes to generate large numbers of different variations of the immunoglobulin, which recognize different antigens. That will be explained later in this video. The pro-B cell expresses CD19 on its cell surface, while the pre-B cell expresses the immunoglobulin M chain in its cytoplasm. Immature B cells express immunoglobulin M, but not immunoglobulin D on their cell surface, while mature B cells express both immunoglobulin M and immunoglobulin D. But this is just an overview. Let's move into detail of lymphopoiesis of B lymphocytes. This is the bone marrow, which is primary lymphoid organ, and this is the peripheral blood, while it's secondary lymphoid organ. This is the follicle of secondary lymphoid organ, while it's the marginal zone of secondary lymphoid organ, and it's the T cell zone of secondary lymphoid organ. The progenitor B cell, or pro-B cell, is first recognizable stage of B cell development. The B cell are self-replenishing cells that express stem cell-associated antigens that are CD34, CD117, and B cell line-specific antigens, such as CD19, CD22, and CD10. Immunoglobulin synthesis begins in the further stage of development. Heavy chains of immunoglobulin M can be detected in cytoplasm of pre-B cell. It has CD19, CD20, and CD22. The next stage of differentiation is called the virgin B cell. It has CD19, CD20, CD21, and CD22. Because it has not come in contact with foreign antigen, immunoglobulin M will be expressed on virgin B cell. The further course of differentiation is antigen-guided. The immature B cells are killed by apoptosis. If their immunoglobulin are bound by autoantigen, presumably presented to them by stromal cells in bone marrow. The other leave the bone marrow at this stage of maturation and then migrate to peripheral blood, called as circulating follicular B cell. having CD19, CD20, CD21, CD22, CD23, CD37, and immunoglobulin M and immunoglobulin D. This cell then enters secondary lymphoid organ. It then migrate to T-cell rich zone of peripheral lymphoid organ, where a process of selection occurs once more. All cells that have not received a survival signal from T-cell will die from apoptosis. The remaining B cells will enter lymphatic follicles. They continuously circulate between bone marrow and secondary lymphoid organ until they meet the matching antigen. This usually occurs in T-rich zone of lymph node or mucosal-associated lymphoid tissue, where B cell develop into proplasma cell. The terminal maturation of B cell into the plasma cells occurs in the bone marrow or mucosa of gastrointestinal tract. Some of the antigen-stimulated B cells migrate to the marginal zone of peripheral organs and differentiate into extracellular follicular B cell. Compare it to the circulating B cell. So extrafollicular B cell are immunoglobulin D negative, CD23 negative, CD39 positive. In contrast to most other B cells, these cells can also react to carbohydrate antigens, called as T-cell independent response, but only generate immunoglobulin M of low affinity. Now, a small fraction of B cells is distinguished by the expression of T-cell associated differentiation antigen, called as CD5+. These B cells are believed to belong to subpopulation that diverges from normal B cell line early on in the course of ontogenesis and colonizes the peritoneal and pleural cavities. This B cell population has been confirmed in mouse. CD5+ are long-lived and self-replenished. They are T-independent, secreting low-affinity IgM autoantibodies, can renew themselves, and undergo no hypermutation. The concept of hypermutation is explained in detail later in this video. This was all about lymphopoiesis of B lymphocytes. If you want to have more understanding, watch our video on primary lymphoid organs and lymphopoiesis. In our next session, we will discuss about the B cell receptor and its structure in detail.